Welcome students to my channel Bio World. In my ongoing topic cell division, this is my third video where I would be starting with the next stage of cell division in mitosis, that is metaphase. In my previous video we studied about the characteristics feature of the stage prophase. Now prophase is followed by metaphase, like prophase and next phase after prophase is metaphase. Now in this phase we need to know the distinguishing feature, what happens during this phase and what are the different characteristics, what are the formation, how the chromosomes they align themselves, what is the condition of the nucleus and the nuclear membrane. All the features has to be studied well. Now you can see on the board I drew two diagrams of the plant cell and the animal cell respectively. Now plant cell is a regular shape size figure, whereas animal cell is oval or spherical. You can understand anyway like that. I have labeled one of them, you can understand, and if it comes in the examination about plant cell, same labeling you have to do it over here. So labeling is must in the diagram. As you see the diagram, you can understand the phase. Whenever you find the chromosomes aligned on a particular plane, in both of these you can understand, in plant cell as well as in animal cell, the chromosomes have aligned themselves on a particular plane. This is one of the most important characteristics that can make you identify this phase in the examination. So whenever you see this image or picture in the paper, you can understand this is metaphase, because this is the only phase where the chromosomes will be aligned like this, in none of the other phases. And we'll go to the different characteristic feature. Now in prophase we found that the nuclear membrane were disappearing, were on the verge of degeneration, they were disappearing slowly. In metaphase we'll find nothing in terms of nucleus, membrane or nucleolus. So we find it starts with spindle apparatus is completely formed. Now in this animal cell we find the spindle apparatus. In the previous one I told you there's no centrioles in plant cell. So there, in place of centriole, there is a microtubule. In the plant cell is a tubulin protein structure that helps in formation of spindle fiber, the thread-like structure that connects the centromere of the chromatids, because it needs to pull them apart in the subsequent phases. We'll be following it. So the first characteristic feature in metaphase is spindle apparatus is completely formed. So we can see from the asters the spindle has come out, the fiber, and has connected itself, has attached itself with the centromere of the chromatid. So each of the chromosomes, each of the halves of the chromosome, that is a chromatid, is attached with the spindle with the centromere. The next one, which I told you already, the nucleolus and the nuclear membrane are degraded completely. That means no way you are finding it anywhere in the image. In the diagram you are not finding anywhere nucleolus or nuclear membrane. It is completely degraded, it's disappeared completely in metaphase. In prophase we found a little shade of nuclear membrane that is on the verge of degrading or disappearing. They were just moving out. In metaphase we are not finding at all. So keep it in mind. You have to, if you are going to draw, if a question comes that draw a stage next to prophase or next to the phase given in the diagram. So if you are asked to draw metaphase, remember in the exam paper you are not going to draw anywhere the nuclear membrane or nucleolus. It is no more found in metaphase. That has to be kept in mind. Third characteristic feature, chromosomes appear clear and distinct with two chromatids in each chromosome. So this is the most important character. This is the phase where the chromosomes could be appear under the microscope very distinctly, very clearly. So you have to keep it in mind if it comes in objective question that name the phase where the chromosome appears distinctly or more clearly. So this is a phase where we find that chromosomes they appear with a full clear vision under microscope. So this is the most clear picture of the chromosome in this phase. Followed it, we have next one, spindle fibers attaches with the centromeres. We understand that spindle fiber has already attached with the centromere of the chromatids, and in the subsequent phase, in the next phase, anaphase, will find them, they are pulling them towards the poles. So they are already ready to pull them. So they have completed their attachment. Next is the most distinct feature of this phase, which will make you identify this phase, that is chromosomes aligned itself or align themselves on the equatorial plane of the cell. So in both of the diagram you can understand that the chromosomes have come on the center of the cell, has come on the equatorial plane of the cell. This has to be in the mind, you cannot forget this. So to keep the stage in the mind, you have to remember that in metaphase the chromosomes are aligning themselves on the equatorial plane, equatorial region of the cell. So they have already come to the mid part of the cell, from where they are going to get separated. They are going to detach themselves. The two chromatids, they are in the chromosome. Now they will be pulled towards the opposite poles in the next phase. So in the next one, we'll go to the anaphase and we'll talk about the characteristic feature of the anaphase stage of the mitosis. Now students, you have to learn these characteristics very nicely. So please jot down this in your notebook or anywhere and learn them out, because these are the characteristic feature you would be asked in the examination when they give you to ask about it. When they give you to like identify the stage, write two characteristic feature on this stage. Along they can ask you to draw them, to label them. Remember that you have to label. The few labeling are most important in these diagrams. You cannot leave the diagram without labeling. It has to be kept in mind. That is the most important thing. And the characteristics, like the fifth one is very important, the second one is very important, the first one, like all of them, the most important I am telling you. So you have to learn each of the characteristic feature for this stage for your own knowledge and study and examination matter. It's very important to learn the feature. Hopefully you liked my video. In my next video, I would be talking about the next stage of mitosis, that is anaphase, with the diagrammatic representation, what happens during that stage and what happens to the chromosome and the chromatid, how they start moving towards the pole, how the furrow starts to take place. These all would be in my next video and in my next particular topic of the cell division. So thank you students for watching this video with patience. So if you have liked this video, please do like, share and subscribe. Do not forget to press the bell icon. Thank you for watching.